Let's move on to lecture objective number 3. I just want to take a quick pause here to see if there are any questions. Let's look at lecture objective number 3. So compare the movement of molecules across the plasma membrane by carriers and through channels. So I want to reiterate here that we're still talking about simple, we're still talking about simple transport, right? We're still talking about having or passive transport. I keep saying simple, I apologize. We're still talking about passive transport. So while it's not simple in that it's not going directly through the membrane, it's still passive. It's still downhill, it's still spontaneous, and it's still going to be releasing energy rather than requiring energy. The difference here is that it's going to require some type of help for these particles to actually get through the membrane. So this is going to happen through a carrier as we think about facilitated diffusion. The carrier is usually a transmembrane protein. And if you recall us thinking about different structures in the cell membrane, we talked about some proteins that go right through both leaflets of the phospholipid bilayer. And so they span the entirety of the membrane. Examples of that would be our facilitated carriers. They're going to have binding sites for specific particles, and the binding is going to happen on one side at a time. So in the example at the bottom here, we have a glucose carrier. And this is a protein, so it's inserted into the membrane. And again, it's fixed and permanently exists in the membrane. It's going to bind glucose on the extracellular surface. So this is the way that we uptake glucose into cells. And so it binds on one side of the membrane. There is a configurational shift or a shape shift of the transporter itself, the carrier itself, that releases the glucose molecule on the opposite face of the membrane, on the intracellular surface. So this is what we mean by facilitated diffusion. The particles can get through because there's a gradient that exists that wants to move the particles according to that chemical driving force. However, because of either the size or shape or charge, the particles cannot go right through the phospholipid bilayer, and so they need help of a carrier that is inserted into the membrane. Now, if we think about the curve of facilitated diffusion, we're going to notice some difference here. The factors that affect the rate of facilitated diffusion are going to be the rate of transport of each carrier. So if we saturate the carriers, we're going to decrease the rate of facilitated diffusion because they're required in order to move particles through the membrane. Also, the individual rate of a single carrier, if we have a really fast movement, high catalytic activity of one carrier, it's going to increase the rate of movement. The concentration gradient is the other one. So again, having a separation in the quantity of particles determines the size or magnitude of the gradient and will also determine the factors, or also determine the rate of transport across the membrane. However, if we look at our curve, we notice that it is not a directly proportional and linear relationship because of the fact that we can max out our carriers. So there's an initial period where the increase in the concentration gradient results in a faster movement or a faster net flux of particles, but then there's a point where that begins to flatten off. And this is where we have saturated all of the transporters or carriers. And so it doesn't matter if the concentration gradient continues to get larger. The limiting factor in this equation is the number of transporters and the individual weight of a single transporter. And so even with a large concentration gradient, there's only but so fast that that rate of transport can be because of the limiting factor being the carriers. Now we can think about a really specific example of facilitated diffusion being our glucose transporter 4, the GLUT4 transporter. These are inserted in the membrane. They're actually made on rough endoplasmic reticula, which are organelles inside the cell where proteins are made. They are packaged in vesicles, and then they are inserted into the membrane, okay? Now, the purpose of the GLUT4 transporter is to uptake glucose to be utilized in our cells, to be utilized in our bloodstream. In diabetes, the lack of insulin, so because diabetics typically have decreased insulin or sometimes they're insulin insensitive, the glucose transporter inserted into the membrane is not as responsive. So the purpose of glucose is to tell the cell to secrete or make more of these transporters and to upregulate the insertion of these transporters into the membrane. Now, again, in type 1 diabetes, there's decreased quantities of insulin. So it's just lower in levels of insulin, which means that there's less of this signaling happening, less of the GLUT4 transporters inserted into the membrane, therefore less glucose uptake. And now there's more glucose circulating in the blood. So this is one of the fundamental problems with diabetes, especially type 1, is that glucose cannot be utilized. So it exists inappropriately and accumulates in the blood because the insulin, the role of insulin is not being is not happening, right? The signaling properties of insulin in order to encourage the GLUT4 transporters and encourage the uptake of glucose, that part is not happening effectively. And so glucose is in the blood, and this is where it can be very damaging. Glucose should not exist just freely circulating in the blood. It can start to affect the fingers, the blood vessels of the eyes, which can lead to blindness. It can affect the kidneys. It can be very damaging as it impacts some of our peripheral tissues. So it's really important for it to circulate in the blood temporarily and then be uptaken into our cells to be a part of glycolysis, etc., and to generate ATP. Let's move on to thinking about channels. So channels are a different mechanism of transport through the membrane. Again, still passive transport, so still obeying a concentration gradient. However, this type of protein is not going to be actively involved in moving the particles across the membrane. They're going to be more just passively standing there like a passageway or a channel in order to move those particles through the membrane. So there are still going to be examples of transmembrane proteins. They're going to be acting specifically, so only certain particles or molecules will be able to move through certain channels, right? They're going to be very specific in that way. But again, not actively involved as a carrier. Carrier is kind of really guiding the molecule through the membrane, especially when we talk about that configurational change, that shape shift. Channels are just standing there and allowing the molecules to pass through. But again, they will be spanning the entirety of the plasma membrane. Some examples of channels are aquaporins. Aquaporins allow water to go through the membrane. So some amount of water can move through by osmosis or just basically diffusion. The majority of water movement through our membranes is going to be via aquaporin channels. Other types of channels are ion channels. So remember we said that the charged particles like sodium, potassium cannot go through the membrane. So the ions need to move through these channels. And there are different sub-classifications of ion channels. There can be leak channels that simply freely move particles through. There can be gated channels, and gated channels will have different mechanisms for opening up those structures. It can be ligand gated, which means something has to bind, or it can be voltage gated, which means the cell has to get to a certain membrane potential in order for that channel to open up. So there can be different gating mechanisms that will determine whether or not these channels open up. And then the third type of ion channel is a bidirectional channel, which means that particles can move, or ions can move rather, in both directions through that channel. Now, the factors that affect the rate of transport through ion channels is pretty much just similar to what we said for facilitated diffusion. It depends on the rate of each individual channel. It depends on the number of channels inserted into the membrane. And then a third factor here is the state of these channels. So with the example of the gated channels that we talked about, if the membrane potential does not favor the opening of these gated channels, then there's going to be zero rate of transport. However, if the membrane potential value that's required has been achieved, the gating mechanism opens up, and then that will increase the rate of movement of ions. Again, I'm going to skip over the questions here just in the interest of time, and we're going to look at the answers when we meet on Wednesday. So again, please write your responses down as homework, as practice from what we discussed so far, and then be prepared to share some of those responses when we meet on Wednesday. We'll begin by reviewing these questions. Let's move on to lecture objective number 3, and we're going to think about the 3 general factors that influence the rate at which a substance, I'm sorry, I think we're on objective number 3, I apologize. So we're thinking about the factors that influence the rate at which substances can move actively across the membrane. So we've talked about passive transport, which is the first part of objective 3, and we're going to move on to the second part of objective 3, which is shifting that focus to think about active transport. And then our fourth objective is really to distinguish between the 2 types of active transport, which are primary active transport and secondary active transport. And then we're also going to think about some specific examples of both of those transport mechanisms. So let's start by recalling the difference between passive transport and active transport. So active transport is non-spontaneous. It requires energy input. And in the instance of transport across a cell, it's going to require a pump, which is what we refer to as a membrane protein that is actively moving substances from one side to the other. We also describe active transport as being uphill, right? If we're going from the bottom of the hill to the top, we're going to have to insert energy in order for that movement to occur. Now, looking at our panel, our example here, here's our membrane across which particles need to be transported. However, the particle in question is not permeable, or the membrane rather is not permeable to the particle in question. So when we think about the movement here, we have this active transporter that's inserted that is going to help the particle get from side A to side B. Now we can think about achieving a steady state. So here we see the movement of particles from one side to the other. Now notice that the movement here is not favoring the concentration gradient. It's actually going to be going opposed to the concentration gradient. So in the instance where we have a lower concentration on side 1 and a higher concentration on side B, so lower on side A, higher on side B, we still have a bidirectional movement through our active transporter because it's not obeying the concentration gradient. It's not passively moving according to the concentration gradient. In fact, in many instances, it's moving against that concentration gradient. Let's think about the characteristics of a pump. It is a transmembrane protein, so we can see it again inserted into the membrane, spanning the full, both leaflets of that membrane. It functions as a transporter and an enzyme. So it has a dual function as far as catalyzing the energy required. So if you remember us saying that active transport requires energy, the enzymatic portion of this transporter is what is catalyzing the energy that's needed to move these particles against their gradient. So it can harness energy that's required for this type of movement. It's going to have specific binding sites, and it is also going to demonstrate saturation in that as soon as the pumps are maxed or saturated, then the rate of movement goes to 0. So here we have our pump, and the example here is actually the sodium-potassium pump. It's one of the famous examples of an active transporter that is seen in many, many instances throughout the body, especially our neurons. This is the basis of the action potential. So here's our sodium-potassium pump. It has 3 binding sites for sodium, and then the other two binding sites here are actually for potassium. So if we think about the pump, this is actually going to move sodium against its concentration gradient. So sodium is typically at higher concentrations on the outside of the cell and lower concentrations on the inside of the cell. So anytime we want to move sodium out of the cell, which is against its concentration gradient, this requires the sodium-potassium pump. We can also see the enzyme portion of that pump here as well. So it will actually catalyze ATP to ADP and use the energy that's stored in that third phosphate to move these ions against their concentration gradient. So this is what we mean by it has a transporter function as well as an enzyme function as well. Now, on that basis, we can think about two types of active transport. There's primary active transport and secondary active transport. Primary active transport is going to result in the hydrolysis of ATP. So again, the main example is the sodium-potassium pump, and the energy that's required to move the particles comes from the cleaving of ATP or the hydrolysis of ATP and the release of the energy in that phosphate. So in the example of the sodium-potassium pump, we have sodium going from the inside of the cell to the outside of the cell, again, against its concentration gradient. So that needs energy. And it binds on the specific sites on the pump. ATP is hydrolyzed, so that's the energy that actually moves these ions from the inside to the outside. Potassium is actually the opposite. Potassium is at higher concentration inside the cell and lower concentrations outside the cell. So in order for us to move potassium into the cell, which is in the direction against its gradient, right, which is going to be from low to high, we need energy in order to move potassium in that direction. So potassium binds on the opposite leaflet. It has its own specific sites. Again, phosphorus is cleaved off, and then that releases the energy to move potassium from the outside of the cell to the inside of the cell. Now, I really want to reiterate that the main feature that separates primary active transport is that ATP is hydrolyzed. We cleave ATP, and that is where the source of energy to move molecules against their concentration gradient comes from. With secondary transport, the energy comes from the movement of another particle that is moving passively. This is why it's so important to think about passive movement as liberating energy, because when we move molecules passively, the energy that's released can be harnessed. It can be taken and used to move another molecule actively against its gradient. So here's another example of that. This example is called co-transport, only because both particles are moving in the same direction. So here's our particle that we're trying to move. We're trying to move it against its gradient, hence the red color of the arrow here. And we're going to link it to a pump that is taking the energy from moving sodium passively. So recall us just describing that sodium is at higher concentrations outside the cell and lower concentrations inside the cell. So the gradient for sodium exists inside the cell. So if we move sodium in this direction, that releases energy, hence the green arrow, and that can be harnessed by this pump in order to move glucose against its concentration. So this is co-transport. The specific example of this is a sodium-linked glucose pump. So a sodium-linked glucose pump simply means that both sodium and glucose are linked together in a single pump that is helping to move both of these particles, one actively and one passively, in the same direction. Now we can think about another example, which is the sodium-linked proton pump or hydrogen pump, which is an example of counter transport. So the same type of mechanism is happening here, which is secondary active transport, we're taking the energy of sodium that is moving passively down its gradient, and we're using that energy by linking these two movements in a single pump, and we can move hydrogen ions or protons against their concentration gradient in the opposite direction. So this one is called counter because we're ultimately moving both particles in opposite directions. Okay, here's a really nice summary table. This can be helpful as you study this. I encourage you to maybe try to block out the columns of each of these areas here and try to fill these in, not from memorizing this, but from really understanding the ways in which these types of transport differ. So simple diffusion, which needs no carriers. Remember, simple diffusion goes right through the membrane. It's going to be down the electrochemical gradient. It requires none of these things. It doesn't require energy, doesn't require transporters. There's no saturation of anything. There's no specificity required. And it's usually particles that are hydrophobic or nonpolar, things like fatty acids, things like oxygen, carbon dioxide. Most of our gases can go right through the cell membrane. When we think about passive transport, so passive transport can also include simple diffusion. I really want to reiterate that, but it can include the other types of transport that require proteins. So this is also going to be down the concentration gradient. That's what makes it a part of being passive, but it is not simple because it requires a carrier. So either an ion channel or a protein carrier is required, which means that there can be saturation, there needs to be specificity, and this is going to basically release or liberate energy. Some of the examples that we talked about are molecules like glucose for our facilitated diffusion, or some of our ions like sodium, potassium for our channels. And then lastly, if we think about active transport, this is going to require energy. So this is moving against the concentration gradient. It's going to be non-spontaneous. And then we can either have it hydrolyzing ATP, which is primary active transport, or using the energy liberated from another molecule moving down its gradient, which is secondary active transport. So for both of these, because there's some molecule, some pump required, we can see saturation, and there needs to be specificity, so specific binding for the molecule in question. Some of the examples of our transporters, primary active transport would be sodium and potassium. So the sodium-potassium pump was one of the primary active transport examples. And then for secondary active transport, we talked about the co-transport, which is the sodium-linked glucose transporter, as well as counter-transport, which is the proton-linked sodium-linked proton transporter, which is counter-transport. So a really nice summary table, really to help you understand and classify these different types of transport. Now, the last thing I want to say here is how all of this really fits together, because this is the basis for what we will talk about when we think about the membrane potential. So if we look at the intracellular and extracellular concentrations, these two solutions differ in the amount of particles dissolved because of these types of mechanisms. So all of the different mechanisms we described, so our sodium, potassium pump here, our leak channels, our ion channels that are moving ions passively. These are the leak channels that allow sodium and potassium to move in one direction passively, and then we have the sodium-potassium pump that moves them in the opposite direction. This is what keeps the relative concentrations of sodium and potassium stable in the extracellular and intracellular fluid, because there's this steady state of ions being pumped in one direction actively, and they can leak in the opposite direction passively. So there's no accumulation of sodium. Sodium is always going to be higher concentration in the extracellular fluid, lower concentration in the intracellular fluid, because of the coordination of both of these types of movement, which is really what we call the pump leak system that keeps the relative concentrations the same. Okay, so the pumping of sodium out of the cell is what facilitates the gradient that allows it to leak in the opposite direction. And these two movements are working hand in hand to keep sodium at a constant concentration.